Hello everyone, my name is Amanda and I'm a third-year student at Rutgers University in the School of Environmental and Biological Sciences and at Douglas Residential College. This summer I worked under PI Dr. Stephen Malin in the Applied Metabolism and Physiology lab. The purpose of this project was to examine how prediabetes phenotypes influence endothelial function in individuals with metabolic syndrome, specifically 22 adults between ages 40 to 70. Individuals qualified for MEDS via the NCEP ATP III criteria, which contains 5 factors: high triglycerides, low HDL, high waist circumference, high blood pressure, and high blood glucose. Prediabetes phenotype, our independent variable, has 4 groups. It was determined via an oral glucose tolerance test where individuals were grouped based on their fasting plasma glucose and post-OGTT plasma glucose. For our control group, normal glucose tolerance, the FPG is less than 100 milligrams per deciliter, and for the impaired fasting glucose, the FPG is greater than 100. The post-OGTT PG measured at 2 hours determines impaired glucose tolerance, which is greater than 140 milligrams per deciliter. And the IFG plus IGT combo group, FPG qualifies for IFG, and post-OGTT plasma glucose qualifies for IGT. While there are 4 PD phenotypes, only 3 were used as independent variables. The IGT phenotype was excluded as it was less common in the data, and this research examined how IGT affects IFG. Endothelial function, our dependent variable, is measured by FMD, which is large artery flow-mediated dilation, and percent FMD is the primary measure of FMD and was calculated via percent change with peak deflation diameter and baseline diameter of the brachial artery. The subject demographics were measured for each PD phenotypic group. These demographics are presented to show that these groups are similar to each other so that we can be more confident that any differences are due to prediabetes phenotypes. They include anthropometric data, which assesses body composition and body size, such as weight, age, waist circumference, and BMI. We also assessed HbA1c, VO2 peak, triglycerides, HDL, systolic and diastolic blood pressure, and heart rate. Graph 1 shows the PG levels at fasting and post-OGTT for each PD phenotypic group. Within each group, the FPG is lower and the post-OGTT PG is higher. This graph shows that PG levels for both fasting and post-OGTT are increasing from NGT to IFG and to IFG plus IGT combo. The FPG and post-OGTT PG measures of each PD group are significantly different from that of each other in every combination. This is shown by the symbols above each bar for each group in this graph. Graph 2 shows the baseline and peak deflation diameter measures of the brachial artery endothelium for the PD phenotypic groups. Within each PD group, the diameter measures for both baseline and peak deflation are very similar, so there were no statistically significant differences between those diameter measurements. Graph 3 shows the average percent FMD for each PD group. NGT has an average of 5.4% FMD, IFG has an average of 6.5%, which is the highest of the 3 groups, and the combo IFG plus IGT has an average of 6.1% FMD. There are differences in percent FMD between PD phenotypic groups, but these differences are not statistically significant. As seen in graph 1, the only statistically significant differences found were between the FPG and post-OGTT PG values. This statistically significant difference was expected because of the design of the grouped phenotypes. These findings are statistically significant, but they are not the main outcome of this project. The main outcome is how endothelial function in MEDS individuals is affected by these phenotypes. So within and among these prediabetes groups, there were no significant differences between baseline and peak deflation diameter. Baseline values between groups are similar as there were no meaningful differences, which shows that the baseline diameter of the vessel is not a confounding variable of FMD. Graph 3 shows the main outcomes of this project, that there were differences in percent FMD between each prediabetes phenotypic group, but not necessarily significant differences. This means that these differences in percent FMD were not due to prediabetes phenotype. Overall, the primary outcome is that there were no significant differences in endothelial function between prediabetes phenotypes and individuals with metabolic syndrome. Based on this data, we can assume that prediabetes phenotype does not worsen endothelial function in folks with a metabolic syndrome. So future research should consist of a larger population of subjects used. Also, since there were no significant differences in macrovascular and microvascular function among the prediabetes phenotypic groups, more research on resistance arterioles is needed as they have a significant influence on blood flow in both micro and macrovascularity. Thank you all, and that concludes my presentation.